All right, welcome to chapter 14. We are going to be talking about um, how cells take the information that is stored in DNA and use that to actually produce proteins. So this ties in with our discussions of gene expression in earlier chapters, right? If you remember that whole term gene expression meant actually producing the protein coded for by a particular gene. And so we've got this chapter split into two parts. In the first half, we'll do just a little bit of background information, and then we're mostly going to focus on transcription, which is producing an RNA copy of the gene, and then the steps that have to be carried out in eukaryotes to process that messenger RNA before it's ready to be translated. And then the second half of the chapter, we'll talk about translation. All right, so of course now we're very familiar with this idea that genes contain these instructions for coding proteins. You need one to get the other, but initially that was not at all clear, and somebody had to figure that out. So we knew that proteins could cause phenotypes. For example, we could isolate this purple pigment from a flower, and we knew that's what was making the flower purple, but we didn't necessarily understand how that was connected to genes. Um, and it was really these two guys, Beetle and Tatum, working on just bread mold of all things, Neurospora, um, that were able to really show that it is genes that code for proteins. And they did this pretty simply. They were blasting the Neurospora they were working with X-rays, which would cause mutations, changes in the DNA. And they found that, well, when they changed the DNA, they were able to change the phenotype of the mold. In particular, they were working with nutritional mutants, so um, mutants that could not produce all the micronutrients that they needed in order to survive. Um, but basically, as long as you understand the idea that they are the ones that kind of establish this link between genes, changes in genes, mutations, and changes in proteins and phenotype. Um, and so they initially called this result, or this hypothesis from their work, the one gene, one enzyme hypothesis, meaning that each gene codes for one particular enzyme that's catalyzing one particular reaction, right? Here's a little metabolic pathway where if we mutate enzyme 1, we would just see substrate A accumulate. Um, Whereas if we mutated enzyme 2 and not enzyme 1, this first reaction would proceed, but then um, this intermediate would just accumulate. And we would get changes in the phenotype of that organism accordingly. It was later realized that, well, um, not all proteins are enzymes. Um, so they changed this to one gene, one polypeptide, and you know, this was really because of proteins like hemoglobin that we've talked about before, where this one protein is made of four different polypeptide chains. So, to make a functional hemoglobin protein, we actually need four different genes, but each one's coding for a particular polypeptide. So, it's sort of splitting hairs, but the basic idea is still sound, right? Genes code for proteins, enzymes, polypeptides. Um, so we're going from point A to point B inside the cells. And this really goes back to what we've talked about. I think we might have even brought this up in the very first lecture of the semester, this idea of the central dogma of molecular biology. And that's that idea that information is flowing in cells from DNA to RNA to protein through this two-step process, transcription and translation. And if we look at that mapped out over a cell here on the right, we can see, at least in a eukaryotic cell, the DNA is inside the nucleus, right? Here's our nucleus. And this process of transcription, basically making an RNA copy of our gene, is happening there in the nucleus. So we make a RNA copy that is eventually going to become what's called the messenger RNA. And then that has to leave the nucleus, and we would translate that RNA into a protein or a polypeptide out in the cytoplasm at a ribosome. So we actually have this physical separation in eukaryotes between those two phenomenon. And you'll notice looking at this cartoon here, we've got a few different types of RNA listed. So, we have our mRNA, messenger RNA that I already listed. You'll see actually that begins life as what's called a pre-messenger RNA, and we have to go through some processing steps that I'll talk about at the end of this lecture. Then you'll notice during the process of translation, we have tRNAs or transfer RNAs. These are molecules that are actually bringing in the individual amino acids that are needed to build the protein, and we'll see those more in the second half when we cover translation in more detail. And the third type is what's called rRNA or ribosomal RNA, and that is actually RNAs that help make up the ribosome itself, and they're really important for allowing the ribosome to form these bonds between the amino acids and build the polypeptide. All right, so of course we have that central dogma. It's arguably one of the most important ideas in modern biology, but there's exceptions to every rule, and there's exceptions to the central dogma. And primarily, I'm talking about viruses. So you might remember we talked about viruses being basically acellular parasites, right? These are RNA or DNA molecules that just need to get inside of a host cell in order to copy themselves, and they do that at the expense of the host cell. So one reason viruses or one way they can serve as an exception to the central dogma is there's a lot of viruses, for example, the coronavirus that's on everybody's mind these days, that has RNA instead of DNA as its genome. So it's basically skipping the transcription part of that central dogma, right? The RNA can just be translated directly into proteins, and we just need to make copies of the RNA, basically of the virus, inside the host cell. And then you've got things like retroviruses. HIV would be an example of a retrovirus that can carry out this unique process called reverse transcription. So normally transcription, as we're going to go through today, is going from DNA to RNA. And it's that first step in the central dogma. But we see some of these viruses have an RNA genome, like HIV, and when that RNA gets inside the host cell, it undergoes this reverse transcription step, so it actually makes a DNA copy of its RNA, and then that DNA, in the case of HIV, integrates into the host DNA. So, if you were to become infected with HIV, as soon as that reverse transcription step is complete, the virus integrates into your chromosome, and at that point you're HIV positive because it's there. It's essentially part of your genome now, and whatever cells were infected. And at least with current technology, we don't really have a way to remove it. Okay, so let's focus in on transcription. As I said, we'll separate out translation for next time. So we're really going to talk about today what's going on up here in the nucleus, transcription and processing primarily. And so there's some similarities to, if we're making new RNA here, there's some similarities to the process of DNA replication. We're making new DNA, but the good news is it's actually a lot simpler. So if you remember in DNA replication, you've got all these different enzymes coming in and doing specific jobs. It's almost basically just a one-man show, one-woman show in this case, one enzyme show, I guess. We have this enzyme called RNA polymerase whose job it is to make the RNA copy. And that makes sense, right? DNA polymerase produce new DNA during DNA replication. RNA polymerase is going to make the new RNA, or the RNA copy of the DNA. And of course, we do need a DNA template to start with, right? We need some DNA to transcribe, and then we also need some nucleotides. And in this case, we would have nucleotides that have adenine, guanine, cytosine, and uracil instead of thymine. But as long as we got those things, we can transcribe. And so here's a little cartoon of RNA polymerase doing its job. And basically the way it works is the DNA that it's going to transcribe kind of gets threaded through this little groove in the enzyme. So it's kind of loaded, locked onto the DNA there. And it will move along the DNA, separating the two strands apart as it goes, just temporarily. So it'll separate them, and then as soon as it passes, they'll stick back together. Um, but while those strands are separated, it's going to read one of the two DNA strands base by base and basically make a complementary RNA copy. Um, and that RNA copy, instead of being stuck to the DNA, so like the RNA primers were during DNA replication, this RNA copy is just going to kind of hang off as a tail, essentially. So RNA polymerase doesn't have that requirement for a 3' end, like DNA polymerase had, so we don't need any special primers. Um, RNA polymerase can act as a helicase, so we don't need a separate enzyme there. So, like I said before, it's really a simpler process. Um, RNA polymerase is precessive. We talked about DNA polymerase having that characteristic too. It can work really, really quickly, but unlike DNA polymerase, RNA polymerase doesn't proofread itself. So it's going to make a lot more mistakes while it's producing this RNA copy. But really, your cell doesn't have to worry about that because RNAs are very unstable and they're just not going to hang around very long. So even if we transcribe this gene and we make some mistakes in this one RNA, um, it may get translated a few times, but then it's going to get chewed up by the cell, and we would have to make a new one that may have less mistakes or different mistakes at any rate. So it ends up not really being an issue. Okay, so let's go through transcription quickly, and we're going to break it into 3 phases. So initiation, how do we start the process? Elongation, which is actually building the RNA copy of our gene. And then termination, how do we know when it's done? How do we stop it? And so we talked about this idea that each gene has its own promoter in eukaryotes, and that promoter is really where RNA polymerase loads and is going to start this process of transcription. So this ties back in with our discussions of gene expression. You remember like operons and so forth. So we have that promoter, and that promoter, you can think of it as really directing the RNA polymerase to the genes. Because if you think of your genome, you have billions of nucleotides of DNA inside every one of your cells, inside each nucleus, and less than 2% of that DNA is actually genes that we want to transcribe. So if you're an RNA polymerase floating around in the nucleus, you have this big challenge, which is, oh, there's so much DNA here. Most of it should not be transcribed. How do I find those genes that I'm looking for? And the answer is it looks for these promoters. And you may remember, it's going to have help from proteins like transcription factors that'll help guide it to those promoters. But in this simple cartoon, we're gonna leave transcription factors out of the equation. But anyway, RNA polymerase will first bind to the promoter here. So in this cartoon, it's the little pink sequence of DNA. Our gene would be over here to the right. Once it binds, the RNA polymerase will separate the two DNA strands from each other, and there will be a specific site there within the promoter that's called the initiation site, which is actually where RNA polymerase is going to get down to business and start the copying process. And then it would simply slide along the DNA off to the right here, unzipping the DNA as it goes and building that RNA copy during the process of elongation, which is what's being shown here. So you can see we've left the promoter behind, right? There it is in pink. And RNA polymerase has moved off to the right. And as it goes, it's just reading, in this case, the bottom strand of DNA here is the template. And it's just, there's just this steady stream of RNA nucleotides flowing into the RNA polymerase. And when the right one, one that has a match with the DNA nucleotide comes into place, it sticks, and RNA polymerase bonds them together, and it moves on to the next one. And we're building this RNA base by base as we move through the gene, and we just get this RNA tail kind of hanging off the side here. And then at some point at the end of the gene, there's going to be a special sequence in the DNA called the termination sequence. And when RNA polymerase encounters that sequence, it knows, okay, I've reached the end of the gene, it's done, and it will literally just fall away from the DNA and letting the new RNA transcript that it made go. So now we have this RNA copy right here of just this one segment of DNA, this one gene. And that's really it in a nutshell. And of course, if you really wanted to dig into it, there are some of these other helper proteins, like I mentioned, transcription factors and such. But that at least gives you the basic gist. So that RNA that we just made in eukaryotes is going to have to be processed. But before we talk about those processing steps, I just wanted to real quickly introduce this idea of the genetic code, because as we start getting into some of these later steps and definitely into translation, we kind of need to have this under our belt. So this term genetic code is basically referring to the order of the specific bases, right? AT, GG, CC versus some other sequence of nucleotides of nitrogenous bases. So it's that order, that sequence that's unique from each person to person, each organism to another. And if you've studied the genetic code before, you've probably heard this term codon. And you can think of codon as the words within these long, like run-on sentences, right? Imagine you open up your book and there's just letters without break, for thousands of pages. But if you start really looking at it, you say, oh, okay, there's a word, there's another word, just sort of back to back. We just left out the spaces. And in the DNA code, the genetic code, these codon words just consist of 3 nucleotides in a row. So the first 3 nucleotides, ATG, would be one codon. The next 3, GCC, would be another codon or word. And each one of those codons specifies a specific amino acid. And I'll show you all the codons here in just a second. But at the beginning of every gene, there's always a start codon, and it is always AUG. So that's that initiation signal for translation. It's basically telling the ribosome, hey, this is the start of the gene where we really need to start making the protein. And then likewise, at the end of every gene, we have a stop codon, and it turns out there's 3 different possible stop codons. You don't have to memorize these. That are telling the ribosome, okay, stop making the protein now. And so we do know the entire genetic code now, and it's sort of interesting to think about how people figured this out. And what they did was, in a nutshell, they were using a in vitro, in the test tube sort of translation system. And what they would do is they could feed in synthetic RNAs and then look to see what proteins were being made, what the proteins look like. And so they started with just sort of monochromatic RNAs. So they would feed in an RNA that was just U, 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 U. So every codon was U, 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 U. And when they look to see what comes out, they're building proteins that are just the same amino acid repeated over and over, phenylalanine in this case. They feed in just a straight AAAA RNA. So every codon is AAA, and that they're getting just lysines. So anyway, by starting simple like this, they realize, okay, UUU codes for phenylalanine, AAA for lysine, CCC for proline, and then they can build on that, right, and start mixing in other nucleotides into these synthetic RNAs and starting to tease that code apart. And so we've got the whole code now, and here it is. So you don't have to memorize this. But just to sort of orient you in terms of what you're looking at here, so this is telling you, okay, these 2 codons, UUU or UUC, both code for the amino acid phenylalanine. all four of these codons, UC, whatever, it doesn't really matter what the third one is, all code for serine, etc. Here's that start codon that I mentioned. Here's those three stop codons in red that I mentioned. So we have this code, and there's really two key characteristics of the genetic code. Well, I guess we could argue three, and I'll come to the third in a minute, but we'd say the code is redundant, but it's not ambiguous. So what does that mean? Well, it's redundant because, as I just mentioned, you have multiple codons that all code for the same amino acid, right? 4 different codons here are all coding for serine, so that's some redundancy. But we say it's not ambiguous because each individual codon, it's always going to code for a particular amino acid. So for example, UCU is always going to code for serine, right? You never find examples where, oh, over here UCU codes for proline. No. It's like if you know that codon and you have this code, you know what amino acid is being specified. I guess the third sort of key characteristic of the code is that it's universal, and as you can see here, nearly universal. There are a few weird subtle exceptions to that rule. But basically what this means is, every organism on Earth is using that same code. Okay, and so you can see a couple of exceptions down here. There's some of the mitochondrial genes are using slightly different codons in some instances, some chloroplast DNA, etc. And there's one weird group of protists, but by and large universal. And this allows some, very interesting modern technologies like genetic engineering. In this weird experiment, this researcher took a gene that creates a green fluorescent protein naturally occurring in jellyfish. So took that gene out of the jellyfish genome and spliced it into the rabbit genome. And what we find is that, that gene produces the same green fluorescent protein in the rabbit cells as it would in the jellyfish cells. And you get the basically fluorescent green rabbit if you put it under the right wavelength of light. And so that's just illustrating the fact that the genetic code is the same between this primitive jellyfish and this more arguably advanced mammal, the rabbit in this case. So we've all got that same genetic code in common. And we're going to get into evolution in some later chapters, but because DNA is the hereditary material and evolution is a phenomenon of heredity, right? It's changes in the genetics of populations over times. This common universal genetic code is really the language of that change. All right, so we went through that process of transcription, and that basically looks the same no matter what kind of organism you're talking about. But in eukaryotes, after we make that initial RNA copy of our gene, there's a few extra processing steps that we have to go through before it's actually ready to be translated into a protein. And so that's what I wanted to go through here. But these modification steps aren't required in prokaryotes. Okay, so this is unique to eukaryotes. And so keep in mind, right, each gene has a promoter, has a terminator at the end. And then what we find in all eukaryotic genes are what are called exons and introns. And so we see these actually in the DNA sequence. And what this boils down to is the dark blue, the exon segments of the gene here are the sequences that are actually important for building the protein encoded for by this gene. And the light blue, the intron sequences here, are just sort of like extra filler DNA in the middle of that gene. And so we're going to have to go through this splicing process to basically get rid of those introns. But we're not gonna actually splice the DNA. We're going to splice that RNA copy of the gene. Okay? So let's see. Let's go ahead here. So this is what it might look like. So we have our gene that has these exons and introns, and RNA polymerase doesn't distinguish, right? It just sees the promoter and the terminator, and it's going to transcribe everything in between those two exons and introns. So it makes the full copy of the gene, right? Even these extra sequences that we don't really need. And so we get our pre-messenger RNA, but before we produce the protein, we want to get rid of those extra sequences. So we will literally just cut and paste. So there are proteins that will come in and make little cuts at the edges of the exons, and then glue those exons together, splice them together, just leaving the introns in the dust. And it looks something like this. So there's these proteins called snRNPs. Those are the ones that actually come in and recognize the edges of the exons. And they sort of loop out, so you'll get one binding to each end of an exon, and then they'll come together to sort of loop out the intron here, and then they make a cut, and severing the intron, gluing the exons together, and then once they're released, holding on to that intron, they just break it down and chew it up. And so you might ask, okay, well, how do these proteins know where the edges of the exons and introns are? And there's these what are called consensus sequences, basically just common sequences that you find at the edge of almost every intron or exon, and that's what those proteins look for, so they know where to bind. Okay, so we do that splicing step, and we'll get into in some later chapters why that happens. It seems kind of inefficient, right? It's like, well, we transcribe it, and then we got to cut and paste. That seems like excessive, and you could argue it is, but there's actually some big advantages to doing that. So we'll come back around to that in some later discussions. So we splice, and then really the only other thing we have to do is just tinker with the ends of the RNA a little bit before it's ready. So to the 5' end, remember each RNA and DNA molecule has a 5' end and a 3' end. So at the 5' end, we'll add what's called this G cap, which is just a modified guanosine nucleotide, basically. The structure of it's not important for our purposes here, but we add this extra structure to the end there, and it is believed that that cap kind of helps protect the messenger RNA from being chewed up by enzymes, so it just makes it last a little bit longer than it otherwise might. And then at the other end, at the 3' end, we're going to add what's called the poly A tail, which is so named because it's just a sequence of mostly AAAs. There's a few U's thrown in there too. This can be an extra 100 to 300 nucleotides there added onto the end. So we get our 5' G cap, and then our 3' A tail. And we think that that A tail also helps stabilize the RNA a little bit, and it seems to be important for helping the RNA get out of the nucleus. So if that poly A tail hasn't been added, then the RNA just stays in the nucleus and would never get translated. Okay, so just to come back to this picture again, at the end here, so we have our DNA in the nucleus, right? RNA polymerase comes in and finds the gene, transcribes the copy into this pre-messenger RNA copy of the gene. We process it with splicing, 5' cap, 3' poly A tail, and then it would be ready to go. So it would actually leave the nucleus through the nuclear pore, and we'd be off ready for translation. And we will talk about that in the second half of the chapter.